pick up where we left off here. So just to review, I always have trouble finding this. Why can't I find this? Play from the current slide. All right, so we had talked about what drugs do. This is an example of an inhibition, a drug that's turning a receptor off. This could be a drug turning a receptor on. So for whatever reason, the substrate is not binding or the receptor is not binding, that's what the disease process is. So we put in the activator, the drug binds here, and now the shape changes and the substrate or the receptor can bind. So again, this is obviously an oversimplification. They can be very specific. The interactions can be very, very complicated, but in general, that's what's supposed to happen. All right, so when a drug is designed, the first thing they have to do is they have to understand the process. Then they have to get a picture of what that active site looks like, where the binding is. Typically, where the drug binds is referred to as the target, the molecule that it binds to. Right? So you have to understand the target. They have to understand the process. They have to know the biochemistry before they can even begin to design a drug, they being the medicinal chemists who work on this material. All right. Let's talk about antibiotics a little bit. So antibiotics typically work on bacteria, and bacteria are very different from human cells. Though the biochemistry can be similar structurally, they're very, very different. So for example, bacteria are what are called prokaryotes. So for example, humans don't have a cell wall. Human cells don't have a cell wall. Mammals don't have cell walls, only plants and bacteria, frankly. This is a cell membrane. We have cell membranes. This is nucleic acid DNA, and they're very, very simple organisms. But for example, cell walls, right? If we designed a drug that could inhibit the cell wall, that wouldn't really affect humans too much. The trick in drugs too is because though organisms are very different, some of the biochemistry is similar. And so if you design something to attack a certain target, it can have cross-reactivity with other molecules that are similar. If it's biologically active, it may be biologically active to humans as well. You know, the idea of what's in the silly idea that was thrown out about, oh, let's just drink some bleach to kill the virus. Well, bleach would kill a virus in a flask. Bleach would kill a virus in your body too, but it would also kill you. And so that wouldn't be a good drug, so to speak. So penicillin is an example of an antibiotic, and what it does is it prevents formation of bacterial cell walls. So penicillin goes in, the bacteria can't make a cell wall, and therefore they just explode, and it kills them. So when they reproduce, they die. That's an example of an antibacterial. Again, we talked about viruses before. Antibacterials, antibiotics, don't work on viruses. So viruses we talked about before. This is a quick review because we're going to talk about this again, just as an example, because right now, during this semester, the medical, biochemical, medicinal chemical communities are working very hard to develop cures for COVID, for this coronavirus, to develop a vaccine. We talked about vaccines a little bit, but we can look at this, and I actually found a really good example, some good examples. So viruses typically contain DNA or RNA. So let's just say nucleic acid, right? They can't reproduce themselves. They have to get into a cell of a host. So the host in case of coronavirus is humans, and they use the host cell's enzymes for all the tools for replication. A lysogenic phase, they can hide there and remain dormant. Okay, lytic phase, like to lyse, that's when you have an active infection and the cells are killed. What happens with the coronavirus is the cells are killed, and then they bleed, the tissue bleeds, and the lungs fill up with blood and other liquids from the immune system, and that's why people can't breathe and they die. Right? So this is a typical viral structure. Coronavirus looks a little bit different, doesn't have these little legs, but there's basically a protein coat and nucleic acid on the inside. Okay, this is what's called this is a virus that infects bacteria. All right, and then again, what they do is they bind to the cell surface, they inject their nucleic acid. In an infection, in a lytic cycle, the viral nucleic acid gets made into viruses, the cell fills up with viruses, the cell pops and dies, and one virus releases many viruses, and this cycle goes on and it gets exponential. All right. In the lysogenic phase, the nucleic acid hides in here like this, and then it can just be activated. An outside stimulus can cause it to go into this cycle. And again, why is it that if I have a viral infection, I feel like crap? Well, I have cells being destroyed that actually have a purpose. So for example, with like the coronavirus, it's going to work on respiratory tissue. And then of course there's all this cell death, and these things rupture, and it can cause, it can cause bleeding into the area. Causes a lot of fluid to build up. The immune system, of course, goes in and tries to clean up the trash, and there's a whole bunch of influx of liquids in there. Right? So antivirals, what they do is they interfere with some aspect of the viral life cycle. So for example, whoops, go backwards. So an antiviral could interfere, whoops, what is going on? An antiviral could interfere with this binding event here. It could prevent the virus from binding. It could prevent the virus from assembling. Some antivirals will prevent the viral nucleic acid from being copied. That would have an effect on the normal nucleic acid as well. But if it's an RNA virus, it has a different life cycle. It can have an effect on the virus reassembling. There's any number of things that can happen. Again, you have to know the specifics of the viral life cycle. In an ideal situation, you would choose a drug that would act on things that are more specific to the virus than on the host cells. Right, so they can interfere with attachment, they can interfere with the self-assembly, any number of things. So I found a really good article. It's very technical. It's from the Journal of the American Medical Association, so obviously a very reputable journal, and it's a review article. So if you click on here, you can read the article. And even though obviously you guys aren't scientists, there's a lot of this stuff that's certainly written at an understandable level, and it really gives, and this was written in April, so it's a month old, and this stuff's been going on. There's been a lot going on, so it's already a little outdated, but it does talk about a lot of the drugs that people have been working on and what their pluses and minuses are, and there's a nice chart in there. So if you want to read a really detailed, and it's a review article means that the people who wrote it looked at a lot of other journal articles that actually have the basic research and pulled everything together into one nice place. All right. So this is, for example, from that article. So this virus, which is an RNA virus, targets cells. So the surface cell protein, okay, the virus has a structural spike on the virus, and it binds to, this is the enzyme that on the cell surface that it binds to, the angiotensin converting enzyme 2. All right. That's an example of what it specifically binds to. Now the other thing too you have to realize is when it's binding to a cell surface, the molecule that it binds to has a purpose. And so if you interfere with that molecule, you can also interfere with what the cells do. And that would be, so to speak, a side effect. So it's tricky. It's not easy. Right? So this is a diagram from that journal article. And the next set of slides when we talk about drug discovery, I'll pick up here. I'll throw this slide in. But this is the coronavirus. This is the enzyme. This is what it binds to. And then it gets into the cell and it does its thing. There's the RNA, makes new virus, and then gets spat out. And again, it doesn't just make one. One that comes in doesn't make one. It makes maybe 1 million of them. All right. And what these are drugs names, and this is what these drugs do. So you can get a feel for how these drugs act. So this one, for example, this prevents viral entry. And these are the chemical names. These aren't like the name on the bottle. This is another drug. It targets this interaction and inhibits the membrane fusion of the viral envelope. This drug prevents the proteins from breaking down. This is one that our president talked about. It inhibits viral entry and prevents it from coming in. All right, so all sorts of stuff like that. So this is actually, if you think about it, it's pretty impressive in terms of how much information we have in such a short period of time. That's because this happened in in 2020, 2019. If this had happened in 1919, this stuff would have not been figured out because we didn't even understand DNA until the 1950s. So that's pretty good. Some other drugs they're working on are targeting turning down the immune system. Anyway, but this is an example of a real-life virus, right? One that's why we're not even in class right now. How it works, drugs that are targeting various processes in its life cycle. All right, so we'll pick up here for drug design. That'll be the last set of slides. And the previous slide, like I said, shows several potential targets. And I talked about knowing the biochemistry of the disease. All right, so that's what we're going to pick up next time. I'll bring some other stuff in from that article. It even has a nice list. That article has a nice list of several drugs that are being tested and where they are in the testing phase, what are some of the toxicities and things that they're working on. And I'll pull that up for the next set of slides. Hope you are all well. Take care of yourselves.